Kanika Gupta, your TA for the session, and today we'll be discussing your doubts, obviously from the lecture, from the week 2 lectures, and also as we always do, we'll first have a recap of what you guys must have listened in the lectures of week 2, and then we'll solve the questions of assignment and some of the questions that I have brought for you guys. So I hope I'm clear to everyone and everyone is able to hear me. If you guys have any doubt, we can discuss it in between also. So there's no need to worry about that. We can do that. But yes, let's start the session now. Okay. Okay, guys. So we'll start the session. Just a moment. People keep on coming. So that is why I'm not getting any notification of people. Mm. Okay. So we'll start. Okay, guys. So in the last lecture, what we did is uh, we in the first week we had a introduction of what is developmental biology, and in the last week's lecture we studied about the differential gene expression, how expression of different gene in different cells or different organs lead to different kind of the phenotype, and that phenotype helps us to make the whole complex organism. That is what the crux of week 2 was, and the mechanisms underlying these different kind of differential gene expressions is what how it happens and what leads to this is what we are going to have a glimpse of in this recap of week 2. Okay, so just a moment, again people keep on coming and I have to add them. Oh, why it is not taking up? Well, let it be, we'll do it later. uh, let's continue where we left off. So, yes. So, genetic core of development is all cells have the same set of chromosomes. That is a universal fact that we know already, that in a single organism, all cells will have same number of same set of chromosomes. Okay, exceptions can be there, but yes, otherwise it is generally universal, that the all cells have the same set of chromosomes. The concept, this is called the concept of genomic equivalence. But what controls the development, the cytoplasms or chromosomes? That is what we are going to see. Obviously the genes is what actually controls these things. Which gene will show what kind of an expression and everything. So we'll see that obviously. uh, so yes, Theodore Boveri is the one who gave the chromosomal theory of inheritance, according to which the chromosomes are equally divided in each cell in an organism, and if the number of chromosome that varies inside the cell, then it can lead to a problem in the growth of that embryo or the zygote, that can lead to the death of the embryo also. That is what the chromosomal theory of inheritance is said. Then these two people, that Nettie Stevens and Edmund Wilson, they told that what leads to the sex determination depending upon the unique chromosomes inside the humans, for example. That is, although the somatic chromosomes are all same in both the genders, but obviously there must be single chromosome difference that makes male male and female female. So what is that? That is the sex chromosomes, that is XX in case of females and XY in case of males. That is what, again, the extension of chromosomal theory of inheritance was, that was proven by Nettie Stevens and Edmund Wilson. Okay. Next was, ठीक है. Then came the germplasm theory, which said that germ cell has all the information for somatic cells also, because obviously a germ cell will have all the information. That is why when it fuses, when they fuse to form the zygote, then only the zygote is able to develop into an whole organism. So that is because this germ cell has all the information which it can transfer to its zygote, and then that zygote will be able to develop into a full-fledged organism. So that is why this germplasm theory said that germ cell has all of the information. So this was a major milestone in biology, and its importance is celebrated in the Darwin's theory of evolution. Then the person Theodore Bovry's experiment with Ascaris embryo that supported the germplasm theory, in which the cell division in Ascaris was observed by him. Ascaris has only two chromosomes, so it was easier to follow during cell division, and then he followed the division in case of the Ascaris embryo, and he found that at last the germ cells possesses all the chromosomes as such, although it got degraded in other cells. But obviously the germ cells, the phenomena of chromosome diminution was there in case of Ascaris, but obviously the germplasm cells carried the exact chromosomes that was required to carry out the information into the next generation. Okay, so that is what he observed. Then next came the other thing that needs to be considered here is that the chromosome diminution is fine for Ascaris because it is not that much complex organism and it is a small organism. But obviously, if we'll talk about humans or if we'll talk about larger complex organisms, then this phenomena is not at all helpful. Otherwise, it can lead to many kind of problems in the developing embryo, and then obviously death of the cells. It can happen because of the presence of unequal number of the chromosomes, or not complete information of the development. Okay, so there's the major objection to nuclear control of development persisted. So the only way left was to show that the nucleus of a fully differentiated cell is capable of generating every other types of cell. So basically the contradiction that developed due to the chromosome diminution that was seen in case of the Ascaris embryo, the conflict was needs to be solved to show that not every organism show this kind of a phenomena. So for that, the only way was to show that the nucleus of a fully differentiated cell is capable of generating every type of cells because it has all the information. But what kind of an information gets activated in that particular cell totally depends upon the environment that is provided inside that cell, because obviously that cell will have a proper environment, proper transcription factors, for example, which will activate those particular genes that are required in that cell. But though it is not necessary that those cell will be required in other cells also, that information will be required in other cells also. So in that case what will happen, it has to have that kind of an environment to get activated. Okay, so Briggs and King were able to make a frog egg develop normally with the transplanted nucleus. So what they did is they took a nucleus from the somatic cells, and then they used that uh, they transplanted the living nuclei from blastula cells into the enucleated frog eggs just to show that it has all the information, and they were able to develop very fine with the nucleus that was taken from the blastula cells because it has all the information. Only what it needed was the environment so that it could activate all of the genes that are required for the development. So that is what these two gentlemen show, that nucleus has all the information, chromosome has all the information. So this is just to show how they did what they actually added the nucleus from a skin cell to egg cell, whatever. So they transplanted the nucleus inside the developing from the blastula into a enucleated frog egg just to show that the development was occurring fine. So, but the nucleus used by Briggs and King came from an undifferentiated cell because in blastula stage, still the nucleus is undifferentiated. The fate is still has to be decided. In blastula stage, it is still dividing. The fate hasn't been decided till now. So it is an undifferentiated cell. So the nucleus will have all kind of information. There is no doubt on that, but it is still not differentiated for a particular fate. So that is why it was able to produce normal tadpoles. Just a moment, guys. This will keep on happening. That's okay. So the development capacity of nuclei taken from intestinal epithelium cells of the feeding tadpole also had all of the information, so it could lead to the development inside the embryo. Okay, so this was what they did just to show that the nucleus has all the information. Every cell has all the information, and that the nucleus of that cell can help in the development inside any other cell also. Okay. Then another evidence for genomic equivalence या फिर that came from this. What they did is they took a Scottish blackface strain of this goat, and another was a Finn Dorset strain. सो इन्होंने एग लिए हैं जहां से ऊसाइड डोनर है, एक से हमने न्यूक्लियस लिया, एक से हमने ऊसाइड लिया. ठीक है. सो एग केम फ्रॉम दिस स्कॉटिश ब्लैकफेस स्ट्रेन और न्यूक्लियस केम फ्रॉम दिस फिन डॉर्सेट स्ट्रेन. व्हाट दे डिड इज they removed the meiotic spindle. Basically they removed the chromosome, they removed the information from this oocyte donor, from this oocyte that they have taken from the Scottish blackface strain. And what they did is they placed this nucleus from this Finn Dorset strain into this, and then led to the development of the egg into a normal organism. So what they found is the embryo transferred to a surrogate mother again through Scottish blackface because oocyst was taken from that only. So it led to the development of a Finn Dorset lamb genetically identical to the nuclear donor. Now what happened here is egg was taken from one strain, nucleus from another strain, and we all know that information is stored inside the nucleus. Obviously oocyte also helps in providing some kind of an environment for the development, but information is still from the nucleus. So although the egg and the developing womb was from the Scottish blackface strain, but the lamb that produced from this combination was a Finn Dorset because nucleus was taken from the Finn Dorset lamb. Okay, so this was what was seen. So this again shows that the nuclei from an it again showed that the nuclei had all of the information, and it is the one which decides what kind of a phenotype will be seen in the organism. Okay. Next thing is again, so cloned animals are not identical because obviously random chromosomal events like crossing over, like random selection of the genes always happens in case of the somatic cells also. And obviously environment is the one which affects some kind of a phenotype of certain genes. So obviously these are two uh, cloned animals, but they are not identical. The fur color is different if you see in both of these. That is because of the random chromosomal events like crossing over and other things that happen in case of the information, the genetic information, which leads to development, some kind of a phenotypic development difference. So, chromosomes direct the development of fertilized egg into an adult, that we have understood. Genetic content of most of the cells are equivalent, that is also what we have understood. Development must proceed by temporarily inactivating parts of the chromosomes rather than losing them. Why this? So that only those gene gets activated that are required at the moment. Not all of the genes get activated at the same time, otherwise it can lead to a chaos and not a proper development of the embryo or the zygote. Okay, so the genetic core of the development we have understood in the initial stage only, the differential gene expression. Genes control the development, that is true, but it is the differential gene expression which actually directs this development, ki which cell will form what and which protein will be expressed in which type of the cells. Okay, if a liver cell is there, and then it should have proteins specific for the liver cell, not the kidney cell, obviously. Otherwise the development will be abrogated and it won't be able to function properly. Okay, so genes can be regulated at different level, like differential gene expression, selective nuclear RNA processing that happens in case of those genes where multiple genes are multiple exons are there in a single gene, and combination of multiple exons form different kind of the proteins. So that kind of a thing happens. Selective messenger RNA translation also happens, and differential protein modification that we will see further in the lecture series. Okay, so first we'll go with the differential gene expression. So, before going into the details of the differential gene expression, first thing that you should understand is that how the differential gene expression is controlled. So, for that, you must understand the structure of a chromosome. So, what is a chromosome? Chromosome is actually an arrangement of histone core proteins wrapped around which is our DNA, which arranges that into a scaffold like structure, which arranges the histone codomains in scaffold like structure, so that a whole chromosome, so whole DNA can be packed inside a chromosome, packed in the form of a chromosome. So if you can see it here, this green part is the one histone core, and the spiral thing that is wrapped around is the DNA, and that then they arrange into different forms to form a complex chromatin, and that chromatin further condenses to form the whole chromosome. ठीक है, तो chromosome is very smaller compared to the whole DNA size. So obviously there must be some kind of a packing that constricts that large size of DNA into a small chromosome. Okay, so this is all a solenoid kind of a structure forms, then that solenoid further complexes to form a chromatin, and then that chromatin further complexes, further condenses to form the chromosome. So it forms a core scaffold kind of a protein. That scaffold kind of a protein helps to keep the chromosome in shape and maintains the structure. ठीक है, so a single unit is called as what? Nucleosome. That means a histone octacore, and wrapped around it is the DNA. The single unit is called as the nucleosome. Then these nucleosomes undergo different kind of an arrangements to form the whole chromosome. Okay, so we're not going into those details that you will understand in your molecular biology lecture if someone if you are taking one. So obviously, the histone core contains these four types of histones. H2A, H2B, H4 and H3. So these are four histone proteins and they are present as doublets. So together they make a histone octacore. Okay? And the linker of the and the DNA that is linking between two nucleosomes, it also has one histone, that is H1 histone. This hasn't been shown here, but this is the H1 histone, which is there to protect the linker DNA also. Okay? So these are the different kind of arrangement, different kind of condensations that occur. So methyl groups get attached to the H3 and H4 histones. So this is something you should know, that methyl groups get attached to H3 and H2 histones, and which leads to the condensation of this nucleosomes, which leads to the condensation of this whole arrangement. Uncondensed nucleosomes, histone tails largely are unmethylated and acetylated. That means the active form, because it is open now. So open forms gives the transcription factor and access to the genes so that they can be transcribed and further function can be performed. Okay, so uncondensed nucleosomes, that means the histone tails here are unmethylated or acetylated, and if the histone tails are methylated, that means the chromosome is the chromatin is condensed and they are packed tightly so that no transcription factor is able to bind there. Okay? So acetyl groups get attached to H2, H3 and H4 histone, and methyl groups get attached to H3 and H4 histones. Okay? Next is these are other different kind of modifications, basically methylation that happens. Not all methylations are inactivating, some are activating also. For example, these are the particular positions of lysines on H3 histones where methylation happens, and then these methylation attract these particular transcription factors, and after attachment of these transcription factors, these particular processes occurs. So for example, if we are talking about the H3 tail lysine at fourth position, it gets attached to CHD1 BPTF transcription factor, which leads to the transcriptional activation. Here it is not inhibition but it is activation. Another is the position 9, where the which upon attachment with the HP1 transcription factor leads to the silence of the heterochromatin. Next is the position 27, which leads to a transcriptional memory. Next is position 38, which leads to a transcriptional elongation. And position 79 leads to the cell cycle regulation. So every methylation has its own role. It is not very random process. It is all very calculated and well informed kind of process. It is not like कि जहाँ पे भी मन किया वही methylation होगी. No, it's not like that. It is very well regulated. Okay, so this is just to show you an arrangement of a gene so that you can know different kind of terms, and these terms are actually very important parts of a gene which makes a eukaryotic gene complete, which helps further in the transcription and then further translation of that particular gene so that it can lead to its particular expression. Okay, so it has a promoter region which has a Tata box in it. Then a Tata box is basically a transcription factor binding site. Then the transcription initiation, it has a 5 prime untranslated region. Then exons, introns and all. Then a 3 prime untranslated region which gets further modified when it forms an mRNA. Then the 3 prime end gets a modification of addition of a poly A tail, which protects this mRNA from getting degraded in the cytoplasm while it is being transcribed. So these are some important parts of a gene which is important for a gene to get transcribed and then translated. Okay, so this is a messenger RNA, as I was telling you. At 3 prime end it gets a modification which is the addition of a poly A tail, and at 5 prime end it gets a modification of addition of a cap. ठीक है, जी कैप यहाँ पे आके लगती है, कैपिंग होती है 5 प्राइम एंड की, और 3 प्राइम एंड पे पॉली एडिनाइलेशन होता है, जिसकी वजह से क्या होता है, जो mRNA है वो स्टेबल रहती है साइटोप्लाज्म में। ठीक है, so again when this mRNA, this mature mRNA is ready after removal of all of the introns, and then addition of poly A tail, addition of the cap on the 5 prime end, then it undergoes translation, it leads to the formation of a whole protein, then that proteins get post translationally modified, and after modification it folds properly, and then it performs its function. So all of the things, all of these steps are necessary before we get a functional protein for a particular cell or from a particular gene. So how these transcriptional factors help, how these are important for different kind of a processes for the initiation of the gene transcription and all, let's see that. So this transcription factor 2D complex is what that binds to the Tata box. As I told you already, it is present in the promoter region of this gene, of a eukaryotic gene. Here it is Tata box. So it is present in the promoter region. So TF2D, transcription factor 2D complex, binds to this Tata box through its Tata box binding protein subunit. Is TF2D also has different subunits. So one of it is TBP, which helps in binding which helps this TF2D complex in binding to Tata box. Okay. Next is the transcription factor 2A, which helps in uh, this TF2D complex, this TF2D transcription factor is stabilized by a transcription factor, another transcription factor, that is TF2A. Okay, so it also performs a very important role. Then TF2B and TF2H join the complex on the Tata box. TF2E and TF2F associate with RNA polymerase 2. So this whole complex forms with TF2D, TF2A, TF2B, TF2H. All of these combine on this Tata box to make a stable complex to stabilize the RNA polymerase, so that it can lead to what? It can lead to a proper transcription and then translation. Okay, so it is a very important process. Next is, then this RNA polymerase, when gets attached here at the promoter region, so as I told you, TF2E and TF2F are there to stabilize RNA polymerase. So RNA polymerase 2 is positioned by TF2B, and its carboxy terminal domain is bound by TF2D, which was there initially on Tata box. Okay, so this is how these transcription factors come together to form a whole complex with RNA polymerase also, to make it a stabilized complex so that a proper transcription can happen. The CTD is phosphorylated. The cytoplasmic domain of this RNA polymerase is phosphorylated by which transcription factor? That is TF2H, and is released by TF2D. ठीक है, RNA polymerase 2 can now transcribe mRNA. Now this whole complex has been stabilized. Now this CTD of RNA polymerase 2 is what? Phosphorylated. Now it is ready to work, and now it can transcribe the gene properly. So before starting the transcription also, the gene has to undergo different kind of preparations. Okay. Now the meaning of different terms, that is, first is enhancers. So what are enhancers? They control the efficiency and the rate of transcription. They regulate the temporal and tissue specific expression. ठीक है, सो ये क्या करते हैं? एक तो efficiency and rate of transcription, ki at which rate the transcription will occur. It shouldn't be too fast, it shouldn't be too slow. So this all can be managed by enhancers only. Obviously enhancers will also get bound with some kind of a transcription factor, some kind of an activating or inhibiting kind of molecules, which will further lead to the control in the transcription rate or the efficiency of the RNA polymerase in transcribing that particular gene. They regulate the temporal or tissue specific expression, whether the gene should be expressed in that tissue or not. That all will be managed by what? That all will be managed by enhancers. Enhancers bind specific transcription factors which help them to to regulate these all things, that which gene will be expressed where and what will be the rate of the transcription. So all it is managed by enhancers, and then enhancers will be managed by transcription factors. Just to identify the enhancer, which enhancer is actually uh, regulating which gene, where it is regulating, where it is not regulating, the enhancer trap is a method that was used initially to understand this thing. So I hope you guys have understood it from the lecture. I'm not going into these details. Reporter genes are the one which help in determining the spatial and temporal specificity of enhancer activity, whether the gene is getting expressed there or not, and at which space, which time. All of this will be controlled by enhancer. And to understand that, for experimental purpose, reporter gene is the one which helps us in that matter. So how it is, so expression of a beta galactosidase using muscle specific enhancer in mouse embryo. This was one of the example. Another is the expression of GFP using the enhancer of lens crystalline in tadpole. So if you can see this green light, this is because of the GFP. GFP is obviously normally not present in the tadpole's eye, but it was first added there as a reporter gene so that we can understand the enhancer, the working of the enhancer in that particular gene. So sometimes conditional knockouts are also made to understand this. Just a moment, I should enlarge this. I doubt you'll be able to see this otherwise. So conditional knockouts are also made in some cases. Generally, recombination is not that much of a random process generally, but if we want to make the conditional knockouts of certain genes, then this recombination is the one which helps. So again, I'm not going into the details, but they have used the recombination process here to understand, to make the conditional knockouts, so that again they can understand the enhancers, they can understand the expression of that enhancer, to study the enhancer basically. Again, the gal for UAS system was also the same, in which they have used gal for transcription factor to understand the enhancer better. This is the PAX6 promoter that they have used. If you can see, this green color is generally not seen. This is because this beta galactosidase, and this beta galactosidase was actually added there to again understand this uh, in front of the PAX6 promoter, to understand the expression of that particular gene and the promoter. So yes, transcription factors work in combination. It is not like transcription factor will work on its own. Otherwise, they just keep on lying in the cell, or they get transcribed, and whenever they get attached to their rightful position, then only they can work and they can direct a gene. So obviously, they work in combination also. It is not only the specific position on the gene, it is also the complex that they form with other transcription factors, so that they can work properly, they can get a proper signal that now it is time to activate, now it is time to inhibit. So all of this information comes from a combination. It is not like a single transcription factor is handling everything. It comes from the combination. It comes from which gene is expressing, where it is expressing, what kind of a transcription factor is there, everything. It again works in a combination. It is not like a particular, only a single transcription factor is handling everything. Okay, so it works in a combination. Enhancers are modulars. but the final activation depends on combinatorial action of multiple transcription factors, as we have seen in case of the gene activation also some time back, in which the RNA polymerase is stabilized by TF2F, while in initially TF2D came there at enhancer, then another transcription factor came, and then that is how they made the whole complex, and then that whole complex led to a safe transcription of that particular gene, and it was directed also that which part of the gene needs to be transcribed. So it again works in a combination. It is never like a single transcription factor is making a whole of the changes. No, it is not like that. It will always work in a combination. Combinatorial functions of transcription factors makes coordinated gene expression possible. Otherwise, तो अगर single transcription factor ही सब changes करेगा, then how will gene know it is that particular time that now how will the gene will be regulated that it is that particular time at which the gene needs to be activated or inactivated. ठीक है? So transcription factors usually contain generally three major domains: the DNA binding domain, the trans-activating domain, protein protein interaction domain. These three domains are the major part of any transcription factor. Okay, so how do transcription factors functions? They recruit histone modifying enzymes and displace nucleosomes. They stabilize RNA polymerase 2 on the promoter, and that is how they lead to what the transcription of that particular gene. Otherwise, the genes are tightly bound around the histone octamer. How will they transcribe, and how will the information be passed on from DNA to RNA, and then from RNA to protein? So it is the enhancers that works in that direction, that helps in opening of the DNA at particular location wherever it is required, and then also helps in stabilizing the RNA polymerase so that it can be transcribed properly. For an example, so induction of an insulin secreting pancreatic cell demonstrate the power of transcription factors. ठीक है? So PDX1, NGN3 and MAFA, all of these transcription factors all of these transcription factors are the one. PDX1 is important for the differentiation of intestinal cells into pancreatic cells. Basically, to form the pancreas, PDX1 is very important. Then NGN3 and MAFA1 are important for these pancreatic cells to secrete insulin or glucagon. Sorry. To secrete the to secrete these enzymes, insulin or glucagon. So this is these transcription factors which work in combination in activating those particular cells, and then allowing them to release their product. Okay, so this is because of these transcription factors. Otherwise, the intestinal cells won't be able to differentiate into a pancreatic cell, and they won't be able to produce these enzymes. ठीक है? Next is the transcription factor cascade. So how are transcription factors themselves expressed in tissue specific manner? So it is because of the cascade that is being followed. For example, MBS activates PAC6, which activates crystalline, insulin, glucagon, somatostatin, etc. So this is a whole cascade. One is activating other, and other is activating another. So this is how this whole process goes on, and that is how they work in a combinatorial manner. ठीक है? The pioneer transcription factors, these bind to their enhancer even if they are on repressed chromosome. So that means they will bind to their enhancer. They'll bind to only to their enhancer, either in the repressed form also. They will be there on their enhancer only, and then this MyoD will come, and it will help in the removal of this inhibitor from PBX whenever the transcription of the gene needs to be activated. Then only the MyoD will come, and then Further, P300 and PCAF will come as transcriptional activators, and that is how it will lead to the transcription of that particular gene. Okay? So, silencers are the one which actually silence any particular gene. So, for example, neural restrictive silencer element NRSC is the one which will restrict some particular genes, so that they won't be expressed everywhere. They will be expressed where they are supposed to get expressed, not in the whole organism. Okay, so those are silencer which will regulate, which will actually silence the gene in other cells where they are not required, except for the ones where they are required. Okay. Next is, ठीक है, methylation of globin genes in human embryonic blood cells. So before this we studied again, we studied about methylation, but that was the methylation of uh, histone tails, histone protein tails. Here we are talking about methylation on the DNA itself, and that too, methylation generally happens in the CpG island area, basically in the area where cytosine and guanine nucleotides are present in excess amount, in a vicinity that makes a whole, in a long stretch that makes it a whole CpG island. Okay, so here methylation is inactivating again, and wherever the CpGs of any particular genes are in methylated, that means that gene needs to be inactivated now, or other, or the transcription at that particular gene will not happen, or at that particular exon will not happen. So that is how the different chains of globin molecule in humans is decided. Obviously, because the fetal blood is different from an adult human blood because of single chain difference in the globin molecule. So this is the different kind of a globin molecule chains is regulated by the methylation of the uh, promoters of that particular gene. So again, as I told you, if if a promoter in the promoter region or near the promoter region, if any kind of a methylation is there, so obviously the transcription factor won't be able to bind in that particular area, and then if it is not able to bind in that particular area, it won't be able to reach the promoter, and that is how the gene will be inactivated. Okay. So again, now here, again the histone methyl transferases, here this methyl group is actually helping the addition of an acetyl group in the 2 minute. Yes, acetyl group in the histone tail again. So this is again a regulation kind of a process. So methylation of a DNA can help in the regulation in the methylation or acetylation on the histone proteins also. This is what they have shown here. So methylation of a CpG island is helping in regulation of the methylation or acetylation or deacetylation on the histone tail of a histone protein. So basically H3 tail. So this is what they have shown here. So again, obviously the methylation, the acetylation, they can be sometimes inactivating, they can be sometimes activating, depending upon at which position they are, in which gene they are, what is the requirement of the cell. So there are many factors that decide the what will be the role of methylation or acetylation, or whether it is on histone tail, or either it is on the DNA. So that totally depends upon the what kind of an expression of that particular gene is. Okay. So obviously the methyl transferases are critically important in modifying any DNA. So de novo methyl transferases are those kind of a transferases which can add methyl group to a strand which does not have any methyl group initially. Okay, so they can do a de novo methylation of any DNA strand. But DNMT1, these are the perpetuating methyl transferases. They can only methylate those DNA strands in which the complementary strand is methylated, so that they have a prior information that this strand needs to be methylated and it has to be methylated here because it has a prior information. But de novo are the one that do not have a prior information, that they do not recognize the methylation that has already happened. They do a de novo methylation. ठीक है. So genomic imprinting is an another thing that needs to be kept in mind. So sex specific methylation and as a consequences sex specific expression. So this basically context to the dosage compensation that happens in case of females, because it has two X chromosomes while male has only one. So genomic imprinting is the one that helps in that dosage compensation in case of the females. Otherwise there will be an overexpression of for some genes in kind of a females in females that can lead to certain complications also. So this is genomic printing that helps in that kind of management. ठीक है. CTCF insulator protein binds to the unmethylated DMR, and then generally DMR is a part of this gene, and this insulator protein helps in uh, this insulator proteins help in preventing the expression of this particular gene in case of dosage composition, while in case of paternal chromosome, if we'll see, the paternal chromosome has this methylated because they have single X chromosome. So in that case it won't be methylated, so the expression will be there and it won't be insulated from the expression. ठीक है. For example this Prader-Willi syndrome and Angelman syndrome. So what happens is uh, particularly in case of chromosome 15, if both the copies, because some genes are there in which both the copies needs to be activated. So this is the case here also. In case of chromosome 15, both the chromosomes, paternal and the maternal chromosomes are activated, so it is showing a wild type of expression. But if a deletion of the maternal chromosome for on this particular chromosome happens, but the paternal is completely okay, it can lead to an Angelman syndrome or disease. And if it happens in case of the paternal chromosome, maternal chromosome is intact, then it can lead to a Prader-Willi syndrome. But if both of the chromosomes show this kind of a deletion, it can be lethal, it can lead to death also. ठीक है. So this is showing you the importance of expression of certain genes on both of the chromosome. It is not like always the dosage compensation will work. ठीक है. So they are just showing the inheritance pattern of the Prader-Willi or Angelman syndrome. So this is what you guys have learned in last week's lecture. I hope you understood it from the videos only. This was just a recap. Still, if you have any question, we can take those up. And if you don't, then we'll move ahead towards the assignment questions. So I'm giving you guys 2 minutes. If you have questions, you can ask, we'll answer that. Otherwise, we'll move towards these assignment questions. I'll just have to. Any questions, anyone from this lecture till now? Otherwise, we'll move towards the question answer part. I hope you guys have seen the assignment. You guys were able to solve the assignment. I'm just hoping you guys didn't face any problem. If you did, we can discuss it now because I'll be taking up first assignment questions and then we'll be going forward with the questions that I have brought for you guys. If someone doesn't understand some biological terms and they are not from the biological background, they can ask those terms. Don't feel shy, you can ask those terms, because obviously it is important to understand the terms also. Biology is all about terminologies. Okay, so you don't have any questions. Let's move towards the question answers of assignment then. So guys, we'll proceed it that way. Everyone will type their answers in the chat box for each of the question when I'll ask. No one will speak that loud so that everyone can participate. Okay. So I expect every one of you to type the answers in the chat box. Okay. So we'll start with the question number one. What does question number one says? What were the conclusions of Bowery's experiment that support the germplasm theory? What did Bowery concluded in the germplasm theory through its experiment? It did an experiment on Ascaris, if you remember. So, everyone type your answers in the chat box, please. I hope you understood the question. Okay, Shalini. Other people, please. Very good, Shalini. Only other people, please, if you guys want to participate. You guys understood the question, right? Question says, what were the conclusions of Bowery's experiment that support the germplasm theory? Okay, I think other people don't want to answer. That's okay. So the answer to the question is chromosome fragments during division is observed on somatic cells but not in germ cells. That means the information in germ cells is always complete. It is never in fragments, but it can be fragmented in case of somatic cells. That is what the experiment on Ascaris embryo showed to Bovary. But obviously, then it was further concluded that all the cells had complete information. Okay. So, next question. Question number two. Experiments by which of the following scientists provide evidence that nuclei of fully differentiated cells are capable of generating all types of cell? Which scientist experiment helped in understanding that? Answer in the chat box, please. And just a hint, it has multiple answers. It is not a single option as the answer. Question number two, answer. Answer for question number two, everyone. Other people. Shali, you're partially right, but think you need to add something more also. It's not the only answer. You're able to see the questions on the screen, right? Okay, so the answer to this question is A and D. So the scientists who gave the evidence were John B. Gurdon and Ian Wilmot. ठीक है. Question number 3. Difference in phenotypes observed in somatic clones is due to. Difference in phenotypes observed in somatic clones is due to. We've seen that in case of the cat. So answer to question number 3 is. Yes, it is both A and D. Random inactivation of X chromosome and environmental conditions. ठीक है. Next question. For the first, we'll go for question number 4. The two families of protein that maintain the memory of transcriptional state throughout adulthood are. The two families of proteins that maintain the memory of transcriptional state throughout the adulthood are 1 and 2. Two proteins are there. So name the two proteins. What are these two proteins? You can answer both of the filling the blanks together also for fourth and fifth. Both of them you can answer together. You guys have done the assignment already or not? Good. So please type the answers here. Question number 4 and 5. The two families of protein that maintain the memory of transcriptional state throughout adulthood are. No one wants to answer, that's okay. Trithorax and polycom. So shall you write? It is trithorax and polycom. Okay, let's move towards next question then. Question number 6. Choose the correct statement. So statement 1 says poly A tail is cleaved from the nascent transcript of RNA before translation initiation. Phosphorylation of residues at the C-terminal domain of RNA polymerase II is a key regulator of transcriptional elongation process. Electrophoretic mobility shift assay is to determine if a pair of proteins interact with each other. Transcriptional factors act in a modular fashion and enhancers act combinatorially. बताइए भाई कौन सी स्टेटमेंट सही है इसमें से. Very easy it is. If you guys have listened the lecture carefully, you'll be able to get that right. Ali, I think you need to check your answer again. Shalini, you also need to check your answer again. इन्होंने पूछा which is the correct statement. You need to tell me the correct statement. Okay, let's see which is the correct statement. So first statement says poly A tail is cleaved from the nascent transcript of RNA before translation initiation, which is very wrong because poly A tail is added to mature mRNA gets mature only after addition of a poly A tail. Poly A tail is there to stabilize the mRNA, so it won't be cleaved off from the nascent transcript of RNA before even translation initiation. So this is a completely wrong statement. Then come to statement number 3. Electrophoretic mobility shift assay is to determine if a pair of proteins interact with each other, which is again a wrong statement because EMSA is done on agarose gel. On agarose gel, we see the nucleic acids, so it is an interaction of protein with the DNA. Okay? It is not for to study the interaction between two proteins. It is between protein and DNA. Okay? So again, this is a wrong statement. Now to statement number 4, transcription factor acts in a modular fashion and enhancers act combinatorially, which is again wrong because transcription factors acts in combinatorial manner and enhancers act as in a modular fashion. So this is again a wrong statement. So the right statement is statement B, which says phosphorylation of residues at the C-terminal domain of RNA polymerase 2 is a key regulator of the transcriptional elongation process that we saw when we were discussing about transcription factor 2b, transcription factor 2f, and etc. Okay, so the answer is statement number B. Now, next question. Which of the following is or are a description of enhancers? Question number 7. Answer everyone. Which of the following is or are a description of enhancers? Okay, so answer is yes, correct. All of the above. They control efficiency and rate of transcription, which is right. They regulate temporal and tissue specific expression, which is again right. And they bind specific transcription factors. So all of these are description of enhancers. Okay. Next question. Question number 8. Mutagenesis using transposable elements is employed in what? Very easy answer, everyone. Yes, it is the enhancer trap. It is the enhancer trap in which mutagenesis using transposable elements is employed in. Okay. Next question is which of the following is the reason why the same mutation cause both uh, Angelman and Prader-Willi syndromes? बताइए क्वेश्चन नंबर 9 का आंसर. Yes, it is sex specific DNA methylation. ठीक है, it is the sex specific DNA methylation which does that. ठीक है जी. Okay, next question. Question number 10. 10 or 11 again, they are together, but you can answer one by one. So, DNA regulatory elements that actively repress the transcription of a gene are, क्या कहते हैं हम उनको? What do we call them? So, DNA regulatory elements that actively repress the transcription of a gene are. Yes, those are silencers. while the boundaries of gene expression are set by DNA sequences called. Boundaries of gene expression are set by DNA sequence called. Yes, it is insulators. So it is the insulators which set the boundary, and gene expression is transcription is actively repressed by silencers. ठीक है? Next question is DNA methyl transferase 1 recognizes dash on strands of DNA. किसको recognize करते हैं? ये हमने क्या बोला था, किस एरिया में जो है मिथाइलेशन होती है डीएनए में? Yes, the answer is methyl cytosines. It is the methyl cytosines which are recognized by these DNA methyl transferases 1. Yes, hemimethylated DNA. You're right, Shalini. So with this, the questions of assignments are done. I hope you understood the question. If you had any query, I hope it got solved also. Still, if you have any questions in the end, if you have any questions for these questions, for assignment questions, you can ask me. Otherwise, we'll be move towards the other questions that I have brought for you guys. Any questions, any confusion in the assignment? Any problem you faced while doing the assignment? No problem at all. Should we move ahead? Okay, let's move towards the questions that I have bought. ठीक है जी. It would be a bit of basic questions also, and it would be a bit of application based questions also, but I tried to bring only theoretical ways more. So differential gene expression refers to what? What does a differential gene expression means? Options are there on your screen. So Shalini, electromobility shift assay is basically whenever a protein interacts with the DNA, you will see a shift in the size of that particular DNA on the gel. Whenever you will run an agarose gel, and if the protein is interacting with your particular DNA, because obviously after interaction the size will increase, it will form a complex, so it won't be able to pass through the pores of the agarose gel that easily that other strands are passing by. So that is electromobility shift assay, which tells you that yes, the protein is interacting with that particular part of the DNA or that particular strand. So this is shift assay just to show you that the interaction is there between that particular protein or and the DNA. Okay. Did you got the Shalini? Okay. So yes, the answer to question number 1, differential gene expression refers to. Rest 20 people do not want to participate in the quiz, or they are just not interested. Whatever, chaliye. So yes, the answer is the phenomena where different cells express different sets of genes despite having the same genome, because obviously genome is same in each and every cell, but the expression of different genes in different cells is different, because obviously it is not like every cell require every protein that will be formed from the whole genome. So this is the differential gene of expression, which tells which cells will need what kind of a expression of what protein. Okay. Next question is, the central dogma of molecular biology describes the flow of genetic information as. What is the central dogma of biology's flow chart? Yes, the answer is again B, that is DNA to RNA to protein. Perfect. Question number 3. Which level of gene regulation is considered the most energy efficient for controlling gene expression? Keyword is energy efficient. Yes, it is the transcriptional control. If you'll control it at the transcription level only, then the cell won't waste its energy first transcribing it, then translating it, and then post translation modification. So all of the steps will be omitted if it will be controlled at the transcription level only. So yes, this is the most energy efficient controlling of gene expression. Next question: a promoter region in a DNA is best defined as. Yes, a DNA sequence where RNA polymerase binds to initiate transcription. Okay? So that is what defines a promoter. Next question. The Tata box is a conserved sequence found in. टाटा बॉक्स की लोकेशन क्या होती है? What is the location of Tata box, everyone? Yes, it is the promoter region of many eukaryotic genes. It is in the promoter region of many eukaryotic genes. Next question. Housekeeping genes are characterized by. Feature of housekeeping genes. Yes, being constitutively expressed in most cells for basic cellular functions, right? Question number 7. Transcription factors are proteins that. Yes, that bind to specific DNA sequences to regulate transcription. You're right. Next question is, the primary difference between cis acting and trans acting regulatory element is. What is the difference between a cis acting and trans acting regulatory element? Question number 8. Answer to question number 8, everyone. Yes, the answer is B. So cis acting elements are DNA sequence on the same molecule, while the trans acting elements can diffuse. Basically, cis acting elements are those elements which are present on the same DNA sequence. Uh, that are present on the same gene, but transacting are the ones which can be transcribed by one gene but can act on another gene also. For example, transcription factors. It is not like the transcription factors are transcribed by the same gene and then act on the same gene. No, it is not always the case. Okay. So, question number 9. An enhancer is best described as. हमने क्वेश्चन रिपीट तो नहीं किया? Just a moment, guys. No. So, an enhancer is best described as what? Yes, a DNA sequence that increases the transcription and can act at a distance from the promoter. Right. Question number 10. Epigenetic regulation of gene expression involves. Epigenetic regulation of gene expression involves, yes, heritable changes in gene expression without altering the DNA sequence. Okay, without altering the DNA sequence. So DNA sequence is not being changed here, but gene expression is being regulated by epigenetic changes. That means some kind of modifications on the genes. Next question. DNA methylation typically occurs at which nucleotide in eukaryotes? Question number 11, everyone. Other people who wants to answer this is a very easy question, I think. Yes, the answer is B. Cytosine in CpG dinucleotides, CpG islands. Okay, the next question is the effect of DNA methylation on gene expression is generally. DNA methylation on gene, what does that generally means? Yes, it is the repression or silencing of transcription. ठीक है? Next, histone acetylation is associated with. What does acetylation on histone means? Question number 13. Histone acetylation is associated with. The answer is relaxed chromatin structure and increased transcription. Acetylation is for activation, it is never for repression. Okay, so histone acetylation means relaxed chromatin and increased transcription. ठीक है? Question number 14. The term chromatin remodeling refers to. Chromatin remodeling refers to. Yes, the ATP dependent alteration of chromatin structures to regulate DNA accessibility. Question number 15. Which of the following best describes alternative splicing? Which of the following best describes alternative splicing? Yes, the generation of multiple mRNA isoforms from a single gene by different exon combinations. So this is what alternative splicing means. The 5' cap structure on eukaryotic mRNA functions to protect mRNA from degradation and facilitate ribosome binding. ठीक है. Okay, so some last questions. MicroRNAs regulate gene expression primarily by, if anyone knows about microRNAs, so otherwise I'll tell the answer. So the answer to question number 17, although I'll tell you the answer. Binding to target mRNAs and inhibiting the translation and promoting degradation. So the answer to this question is binding to target mRNAs and inhibiting the translation to promoting degradation. ठीक है. Question number 18. The lac operon in E. coli is an example of. What kind of a regulation? Question number 18, everyone. Yes, it is an example of both positive and negative regulation of gene expression. ठीक है. Question number 19. A repressor protein functions by. Yes, binding to operator sequence and blocking transcription. ठीक है. Next question, question number 20. The concept of combinatorial control in gene regulation means. इस पे आज हमने डिस्कस भी किया है. So what does that means? Question number 20, everyone. Yes, multiple transcription factors work together to regulate a single gene. So that's it. These were the questions that I've brought for you guys. If you have any confusion in any topic, you can ask me. A microRNA, Shalini, these are basically, I'll just give you a short introduction. So microRNAs are basically the small RNAs which are generally to regulate obviously the gene expression. These are the complementary RNAs to some specific genes which are short, very like, if I'm not wrong, 20 to 25 nucleotide long, and they bind to that specific region in the gene so that the expression of that gene can be inhibited. So these are generally, we can say, complementary RNAs, short RNAs for some specific genes to regulate their gene expression. How they are formed, and that is a very detailed process that is something we're not discussing here, but these are the short RNAs which are there to control the gene expression. Any other question, anyone? Yes, these are for transcriptional repression because they'll bind to that area of a they'll bind to particular area of a gene, and transcription won't be able to proceed further. Sorry, not specially for transcription repression, translation repression. So sorry, I just got confused in translation repression. So they will bind to the mRNAs, not gene particularly, but mRNAs, and they'll be complementary to mRNA because mRNA is what, a single-stranded single-stranded structure. It is not a double-stranded structure. So these particular short RNAs are complementary to mRNAs. So because DNA has a complementary strand, so where will they bind? They'll bind to mRNAs. So transcription will happen, but translation won't. So these are for the translational regulation. Any other question, anyone? If you don't have any questions, then you can leave the meeting, guys. Thank you for joining. Hmm.